Hi everyone, we are excited to present our study titled Deciphering Unique and Shared Interactions Between the Human Gut Microbiota and Oral Antidiabetic Drugs. The study has recently been published in the iMeta journal. Numerous studies have demonstrated the bidirectional interactions between oral antidiabetic drugs, OADs, and the gut microbiota in patients with type 2 diabetes. However, considerable disparities persist in previous reports, possibly due to the variations in ethnicity, sample size, and metagenomic methodology across studies. A comprehensive evaluation using consistent pipelines is clearly warranted to understand bidirectional interactions between OADs and the gut microbiota, as well as their potential impacts on drug efficacy. This study encompassed 470 type 2 diabetes patients from 6 clinical trials, including 5 publicly available studies, and an open-labeled, single-arm clinical trial conducted in the present study. We revealed that both acarbose and berberine treatments significantly reduced species-level richness and Shannon index and altered community composition. By contrast, such changes were not observed following administration of metformin, vildagliptin, glipizide, or placebo. At both the species and functional levels, unique and shared alteration patterns were observed among different OADs. At the species level, both acarbose and berberine significantly increased the relative abundances of multiple firmicutes members. Conversely, a reduction in relative abundance of bacteroidetes members was observed in response to acarbose and vildagliptin treatments. Additionally, both berberine and metformin treatment induced an increase in the relative abundance of Escherichia coli and a decrease in the relative abundance of Clostridium bartlettii. At the functional level, both acarbose and berberine significantly increased the relative abundances of the gene encoding 7-beta-hydroxysteroid dehydrogenase while concurrently reducing the relative abundances of genes involved in secondary bile acid transformation. Except for glipizide, all other oral antidiabetic drug increased the abundances of genes involved in glycerolipid metabolism. We found that acarbose combined with a high-fiber diet led to not only a significant increase in butyrate producers but also a significant reduction in the relative abundance of the 7-beta-HSDH gene, exhibiting distinct patterns to the acarbose alone groups. We further revealed that baseline gut microbial features explained 12% to 39% of the response variance in HbA1c, 12% to 35% of the variance in FPG, and 6% to 30% of the variance in HOMA-IR across OADs groups. Notably, higher baseline abundances of Bacteroides spp. Tended to be associated with low responders in the vildagliptin group, but high responders in the berberine group. Furthermore, specific baseline features were consistently associated with antidiabetic effects across oral antidiabetic drugs groups, including associations between higher abundances of BSH genes and greater treatment improvements in metformin and acarbose group, and those between higher abundances of glycerolipid metabolism genes and greater HbA1c reduction in metformin and vildagliptin groups. In conclusion, we conducted a comprehensive analysis on 1,045 metagenomic samples and revealed extensive bidirectional interactions between oral antidiabetic drugs and the gut microbiota in patients with type 2 diabetes. Our findings revealed both unique and shared oral antidiabetic drugs-induced microbial alterations, uncovering complex interactions between acarbose, a high-fiber diet, and the gut microbiota, and highlight the intricate relationships between baseline gut microbiota and therapeutic effects impacting on metabolic health.
Finally, we would like to express our gratitude to our team for their dedicated efforts in this study. We also extend our thanks to the editorial team at iMeta for their full support in the publication of our paper. And thank you all for your attentive listening.